It's my great pleasure to congratulate and welcome to the stage Dr. Madeline Lancaster.
Good evening, and thank you for this immense honor. When I found out that I would be this year's laureate in life sciences, I was truly surprised. You see, when I started in science, I could never have predicted that I would be here today. Halfway across the world, where I started, which was in the western United States, I grew up in Utah, a beautiful place full of hiking and camping, river rafting and skiing. Here is actually where my mom and my stepdad got married, under this beautiful natural altar, the Delicate Arch. All of this beauty made me appreciate the natural world, and I wanted to understand it better. And so growing up with my father as a biochemist, here he is as a budding scientist, and my mother as a psychiatrist, it may have been fate that the natural object that would fascinate me most would be the human brain. So our minds are incredible. We are able to learn to communicate, not just simple facts, but abstract ideas. We can recall the past and plan for the future. We have imaginations and creativity. But when I started my research career, I realized that to understand the brain, it's not enough to just look at it. It won't reveal its secrets so easily. To truly understand how something works, you have to understand how it's built. And make no mistake, as incredible as our minds are, like every other part of us, they are built. And that idea is how I fell in love with development. That a few tiny stem cells early in development can make such an elaborate and beautiful structure is truly awesome. I wanted to know how this worked, so I took a huge leap, both scientifically and geographically. And I moved to the beautiful city of Vienna. Here I am with friends. And I began working with Jürgen Knoblich, a brilliant developmental fly biologist, on something a bit crazy, building a brain. Now, the human brain is not simply an enlarged mouse brain. Our brains are also several orders of magnitude more complex. That means that not only are there more cells, but that those cells have many more connections, and those connections create much more elaborate networks. But that immense complexity is both a gift and a curse. It gives us our unique talents, but it also makes our brains more fragile, so that even relatively mild abnormalities have devastating consequences. Can we study and truly understand a condition like schizophrenia, marked by hallucinations like hearing speaking voices and delusional behavior in a mouse? The answer is we can't, not completely at least. So scientists need human models as well, models that can start to reveal details that are specific to us. So I created a model. So first I needed to figure out the ideal conditions under which I could take human samples and then turn them into stem cells. So for example, what kinds of nutrients do they need? What should the temperature of the environment be, and how much oxygen do the cells require? And so once I figured that out, I was able to grow some of those incredible pluripotent stem cells. On the left is an image of what these cells look like under a microscope. Without much further input from me, these stem cells actually self-organized and grew into embryoid bodies, which are little balls of cells that sort of start to spontaneously develop and form different cells of the body. By adjusting the composition of the culture bath, I was then able to push them to become what's called neuroectoderm, the primitive tissue that becomes the brain. Following the neural tube developmental milestone, the tissues become a bit more recognizable. With a little agitation, they grow into what we termed cerebral organoids, or brain organoids. They aren't an organ because they're not a fully formed human brain. Instead, they're organoids, mini models of the brain. And so here is an organoid injected with dye. That's beautiful, right? So now I know what you're thinking. That doesn't really look like a brain, but it actually does. So you see each of these bulges coming off. Each one of those is like a little developing cerebral cortex. In this one, the blue dye you see allows you to see that each one of those little lobes has a space full of cerebrospinal fluid, just like an actual developing brain. And here's one where I've cut it open and stained it for different cell types. So in pink, you can see the progenitor cells, those cells that will make neurons, and in white and green around the edges, you can see the newborn neurons themselves. And when you compare an organoid to an actual developing brain, you see that those lobes look a lot like the real thing. So now that we have created this model of the human brain, can we use it to study brain disorders? We can, and I want to provide you with some of the ways that we and others are starting to do that. So one of the first disorders we were interested in studying is a condition called microcephaly. In this disorder, babies are born with abnormally small brains. I'm sure everyone here remembers when scientists discovered the link between fetal Zika infections and microcephaly, following the Zika outbreak in Brazil in 2016. Microcephaly occurs when something goes wrong during fetal development, but we still don't understand exactly how it happens. As you can see, the changes in the human brain, in human brain development are profound. However, scientists have not been able to model this disorder in mice. So even when you introduce a mutation that causes the disorder, the mouse brain is only modestly affected. So we asked, if we created brain organoids using stem cells from people with microcephaly, could we develop a better model for this disorder? We did, and we were successful. Brain organoids made from a person with microcephaly ended up dramatically smaller than organoids made from donors with normally developed brains. And because the organoids mimicked for the first time what was actually happening in patients, we were able to use them to uncover why they were physically smaller. And it turns out that the mutation leads to premature formation of neurons, so that the progenitors get used up too quickly. Scientists around the world are now using this method to create organoids from patients with microcephaly and use them to study this disorder. Researchers are also using brain organoids to look at a wide variety of human neurological and mental health disorders. It is now possible to collect samples from patients with autism spectrum disorder, schizophrenia, and Alzheimer's disease, to create organoids and then to study them. And so where do we go next? So we're now making more and more advanced forms of brain organoids. This is an area inside your brain called the ventricular system. Everything highlighted in yellow is actually an empty space that is filled with a fluid called cerebrospinal fluid, or CSF. There are specialized cells inside the ventricles called the choroid plexus that produce CSF. These cells also constitute a really important barrier in our brain. This barrier is among the least studied structures of the central nervous system because it is so difficult to model. Recently, we created brain organoids that can produce CSF. You can see that the lobes on either side look a bit like ventricles with large balloons filled with fluid. And that is actually CSF being made by the organoids. Understanding how this part of the brain works is vitally important because these cells act as a barrier between our brains and the rest of our bodies. It is how our brains receive nutrients, and it's also how our brains receive drugs. Pharmacologists and chemists come up with a lot of great ideas for drug therapeutics, but it's often the case that those drugs can't get into the brain. They can't cross the protective barrier of the brain. So this is a model that we can use, finally, to study the system. The CSF-producing cells may also be the reason some patients experience neurological symptoms when they are infected with COVID-19. Using brain organoids, my lab identified that the CSF-producing cells in the brain are highly susceptible to the virus that causes COVID-19. When the cells are infected, the barrier that protects the brain actually breaks down. We're now also finding ways of engineering brain organoids so that the neurons within make long-range connections, just like the actual human brain pictured here. This is an image taken in an MRI machine showing neuronal fiber bundles. These are long connections that allow different parts of your brain to talk to each other. And here is an organoid with a very similar pattern of long-range projections. We can even connect organoids to living muscle tissue and observe functional connections. You can see here as muscle twitches when we stimulate an organoid that we've cultured together with spinal cord and muscle tissue. This model can be used in the future to study disorders like epilepsy, multiple sclerosis, ALS, or Parkinson's disease. And finally, we're creating new ways to study not only humans, but non-human primates as well. On the left is an organoid made from human stem cells, and on the right is an organoid from gorilla stem cells. You can see how they are both visually similar, but also quite distinct. Studying all primates, human and otherwise, has enormous potential to show us how we evolved, where we as a species came from, and what, on a molecular level, has helped to make humanity the singular species that it is today. And so with that, I'd like to thank the Blavatnik Family Foundation and the New York Academy of Sciences for their support of fundamental discovery in the life sciences. I also want to thank my supportive family, my mentors and colleagues over the years, and the people at the heart of it all, the brilliant scientists in my lab. Thank you.